Good afternoon, students.
Good afternoon, ma'am.
Many have exams, I think. So today they're going the class. So what's that we were doing in the previous class? Purvi, can you please give the recap of everything what we learned in the previous class? Yeah.
We were doing body fluids and circulation.
Sorry.
Body fluids and circulation.
We finished body fluids and circulation, right? And then we took up
Breathing.
respiration, cellular oxidation. Do you remember?
Oh yeah, same thing.
Yeah. So what we did. So I think even Krebs cycle we finished. Did we finish Krebs cycle?
Yes, ma'am.
Yeah. Can you give the brief account of everything? Akash, you were not there. Just a minute. Yeah. I'm sorry. So these kids were making noise, so I had to tell. Yes. Purvi, you were giving the brief account of what we learned in the previous class. Akash, so you were not there. Please listen.
So we started off with glycolysis.
Yeah.
Which is a common pathway
before going to that. Yeah. So for Akash's sake, I'm telling you again. So we did aerobic and anaerobic respiration. So hopefully you know that aerobic and anaerobic. So which you have learned in your 10th standard also. So how the glucose molecule is broken down to 6 carbon compound is broken down to 3 carbon compound. So that happens in irrespective of aerobic or anaerobic. So then we did the glycolysis. So that is glycolysis is the breakdown of the glucose to the 3 carbon compound, that's pyruvate. So that happens in cytoplasm. So irrespective of whether it's an aerobic or anaerobic, it happens, it takes place. So what is Vihan, can you please give me the balance sheet of glycolysis quickly? So balance sheet glycolysis, the 10 steps has been given in your textbook, Akash. So also I would be uploading the video related to that, what we did it in the previous class on Class Pro. So you would get the access to that. So please watch the video so that you can connect it, whatever we are learning today. Yeah, Akash. Yeah. Yes. So now I want, so that's what we did in the glycolysis. So what is the balance sheet? Can you give the balance sheet of glycolysis, Vihan? Balance sheet to the glycolysis. Energetics to the glycolysis or balance sheet to the glycolysis, whatever you can call it as. So how is the balance sheet with respect to the glycolysis? Or else the net equation of the glycolysis. Can you give the net equation to the glycolysis, Purvi?
Ma'am, the net ATP form.
Yeah.
Ma'am, I think 2 is used in the investment phase and 4 is produced in the payoff phase.
Yes, glycolysis has two phases. That is investment phase where ATP is used and payoff is where ATP is released, given out or produced. Okay. So in that case, so how many ATP is used? Overall, 2 are used, 4 are produced. So the net ATP produced is 2. Okay, you have invested 2 and got 4, but net profit is 2. So net production is 2. So that's what the net ATP equation. So overall, the net equation of the glycolysis is C6H12O6. So if I ask you to give the chemical equation for the glycolysis, it is glucose C6H12O6 plus 2 adenine diphosphate ADP and 2 inorganic phosphate plus 2 NAD, that is nicotinamide adenine dinucleotide, which is a vitamin derived coenzyme. Okay, so we get pyruvate. So that's a 3-carbon compound. So what is the formula for pyruvate? CH3COCOOH, 2CH3COCOOH, right? C3H4O3, which is, so you get 2 of that and 2 ATP, 2 NADH and water. Okay, so when NADH is formed, 2 NADH and H plus and H plus, 2 H plus, and water is also released. So that's why when we breathe out, so we get water. So now what's the fate of this pyruvate? This is your subjective question that could be asked in your exam. So what is the fate of this pyruvate? So what happens to the pyruvate when it is formed 3-carbon compound? How is it utilized? So this pyruvate is used to give the ethyl alcohol, we call it as, so what's the fate of pyruvate? Pyruvate has 3 fate. Let me share the screen so you will know. Can you see the screen? Akash, are you able to see the screen now?
Yes.
Yeah. So what is the fate of pyruvate? Pyruvate has 3 fate. So that is anaerobic. So that's lactic acid fermentation, alcoholic fermentation, and the pyruvate which enters in the form of coenzyme A into the mitochondria, into the matrix of mitochondria where the Krebs cycle occurs. So these are the 3 ways of pyruvate, fates of pyruvate. So if pyruvate is broken down to give energy in the adequate supply of oxygen, so C3H4O3, okay, so this much is the pyruvate in the presence of lactose dehydrogenase. So whatever the NAD that's used up during glycolysis is used up here to give, see, that is dehydrogenase, that's why we say, and lactate is formed. So this is one fate. So this happens in the muscle cells. And this is the alcoholic fermentation which happens in any anaerobic bacteria, anaerobic yeast. Yeast is a classic example, which is also commercially Saccharomyces cerevisiae. That is the scientific name of yeast. And here again, so there is decarboxylation. So decarboxylation is not there here, and you find it here. So decarboxylation, carbon dioxide is still given out. So, and here acetaldehyde is formed, the intermediate product is formed, and here in this case, in lactic acid fermentation, you do not find any of this acetaldehyde form, I mean intermediate form. So acetaldehyde is converted to ethanol. So weight is converted to acetaldehyde and acetaldehyde is converted to ethanol. So that is alcohol fermentation. So that's why we call it as alcohol fermentation. And this is what we get in brewing industry. So now this is the alcoholic fermentation. So another, just like that I have shown. So the overall reaction is 2-pyruvic acid. So every time from one molecule of glucose, you get 2-pyruvic acid, and Carbon dioxide is given out. You can see this. So 2 carbon dioxide molecule is given out. Acetaldehyde formation and acetaldehyde is converted to ethanol. So if there is more than 13% of alcohol that is formed, then you know that alcohol is fatal to the microorganisms. So that's why we have alcohol in the whatever Savlon, Dettol, or you take the sanitizer, what is there in that? So in alcohol. So any kind of surgical instruments are also cleaned with the help of alcohol. So you know that alcohol is detrimental to the microorganisms. So hence 13% and more than that alcohol is formed in, so it becomes fatal for the yeast. Okay, so now let's take some of the questions moving ahead with the critical Krebs cycles. We are done with that. So I'm just giving the recap because Akash, you were not there in the previous class. So with respect to that, so come on, solve all these questions now, right now. Which of the following processes carbon dioxide is not released? Yes, go ahead with your answers. In which of the following processes carbon dioxide is not released? Yeah, can you give me the answer?
Is it D-lactate fermentation?
So aerobic respiration in plants, anaerobic respiration in plants, in animals. So that is carbon dioxide is not given out in lactate fermentation. Just now we discussed this. So the energy releasing metabolic process in which substrate is oxidized without an external electron acceptor is called an energy releasing metabolic process in which substrate is oxidized without an external electron acceptor. What's your answer?
Is it photorespiration?
Do you get?
Oh, fermentation.
Exactly, fermentation. So in alcoholic fermentation, what is the answer? In alcoholic fermentation, come on, give the answer. Yeah, read out the question, read out the options.
Triose phosphate is the electron donor, while acetaldehyde is the electron acceptor. Triose is the electron donor, while pyruvic acid is the electron acceptor. There is no electron donor. Oxygen is the electron acceptor.
Yes. So what's your answer? In alcohol fermentation, triose phosphate is the electron donor, while the acetaldehyde is the electron acceptor. Triose phosphate is the electron donor, while pyruvic acid is the electron acceptor. So there is no electron donor. Oxygen is the electron acceptor. So which one is your answer? This is the latest question.
There is no electron donor.
Where? What's your answer, Bihan? Why Bihan is not talking today? What happened to Bihan?
Can you hear me? Can you hear me?
Yeah, I can hear you.
Yes, ma'am. So the 10th question, right?
Not the 10th question. It is 8th question. I'm sorry, 9th question. Just now we discussed in alcoholic fermentation, which of the following would happen? Which one is the electron donor? Well, acetaldehyde is an electron acceptor. Do you say yes? So just take a look. Pyruvate is converted to acetaldehyde. So then acetaldehyde is converted to ethanol. So here carbon dioxide is released, and whatever the NADH and H plus is the energy precursor that's formed during glycolysis is used up to convert the acetaldehyde to ethanol. Right. Which one is the answer?
A.
What do you think is the answer? I want answer from you guys only. You have to think now. Purvi, Vihan,
Triose phosphate is the electron donor while acetaldehyde is the acceptor.
acetaldehyde is the electron.
Acceptor.
Why? Why did you say that?
Ma'am, it takes an electron and it gets converted to ethanol.
Yeah, so that is when proton is lost. Do you say electron is lost?
No.
So this is your chemistry knowledge. So that's why chemistry is also checked here. Akash and Vihan, what's your answer? Why no one is answering? What have you understood about fermentation? So I'm showing the screen. Can you see the screen? Okay, let me show you here. See, alcoholic fermentation, what is the case? Just take a look. Can you see the screen?
Yes.
Akash, Vihan, Purvi.
Yes, ma'am.
Yes,
Yeah.
ma'am.
So what's your answer there? So now take a look here. So pyruvate, this is decarboxylation. Carbon dioxide is given out and pyruvate is converted to acetaldehyde, and acetaldehyde is further converted to ethanol formation. So what's happening here? NADH is utilized. So that is dehydrogenase, that is lactic acid dehydrogenase, and here reduction takes place. I have given you the clue here. So what is converted? So I think Purvi told the answer. What's the answer, Purvi? Again, read out the answer.
Triose phosphate is the electron donor while acetaldehyde is the electron acceptor.
So why did you say that? That explanation only very important.
Ma'am, on your screen it said reduction. So that is, reduction is addition of electrons. So that's why acetaldehyde is the electron acceptor.
Sorry?
Ma'am, it's reduction, right? Acetaldehyde gets reduced. So reduction means
Correct.
acceptance of electrons. So that's
Very correct. Very good. So acetaldehyde is reduced to alcohol. So what happens? This we have done in the previous chapter also. So that is G3P. So that is in the previous chapter when we were learning Calvin cycle. I told you reversal of glycolysis, where acetaldehyde, aldehyde is converted to alcohol and alcohol is converted to aldehyde on what basis? So this is your chemistry, you would have learned that. When you add hydrogen, what happens to aldehyde? acetaldehyde is converted to ethanol by accepting electron. So, are you understanding, all of you?
Yes, ma'am.
Yes, ma'am.
Other students? Triose phosphate. Triose phosphate is which one? Which one am I calling triose phosphate here? What is triose phosphate, Purvi?
Is it pyruvate?
Yes. Triose phosphate, triose. Pyruvate is triose. Glyceraldehyde 3-phosphate. G3P, we have come across that. So it is converted to, so here triose phosphate is the electron donor, while acetaldehyde is the electron acceptor. So in this case, so pyruvate donates it, and so acetaldehyde. How? By losing one carbon dioxide. Right? Acetaldehyde now will have 2 carbon. So that's why C2H5OH is ethanol. So now C2, so it loses oxygen also. So hence it is decarboxylation happens there, and it accepts acetaldehyde is reduced. So this is the concept there. So the next question. So who will do the next question? 10th question, the end product of, yes, Vihan, read. Read, Vihan.
I can't see your screen.
Why?
Ma'am, I think you have stopped sharing. I can't see the screen.
Stopped. Oh, I'm sorry. Now,
Ma'am, yes, ma'am. No, it is.
yeah, please read. Yeah.
Ma'am, ethanol and CO2.
So this is this energy releasing is fermentation. The end product of, yes, ethanol, C2H5OH. Right. Which of the following products are aerobic respiration from yeast? What do you get from yeast?
All of these.
So from anaerobic respiration from yeast, so you get beer and all of these. So when yeast ferments glucose, the products obtained are
Ethanol and CO2.
ethanol and CO2. Yes. So now what happens to the pyruvate? So as I told you, it's not alone glucose that's the substrate, respiratory substrate. Respiratory substrate can be lipids also, proteins also. So what happens? So oxidation of the pyruvate. So this is what? So this is by pyruvate dehydrogenase. And pyruvate dehydrogenase, I told you, is the most complex enzyme or the protein that's present in our body. So in plants' body, so which has magnesium, TPP, thio. So it has 60 subunits. So which is the most complex respiratory enzyme means you have to say pyruvate dehydrogenase. So this pyruvate dehydrogenase, pyruvate loses carbon dioxide. Keep counting the number of carbon dioxide that's lost in every step. Okay. Is converted to acetyl coenzyme A. So as a coenzyme A, and we had done with this also. Do you remember?
Yes, ma'am.
Do you remember we completed this also? And let's do it fast. This one. So let me enlarge the picture. See, I have added so many questions for you guys. So you need to do more and more questions. So how to bring it down the view. Is it too small for you guys?
No, ma'am.
Just tell me. Yeah. So now one of you explain this. Yes, Purvi, go ahead. So acetyl coenzyme A. So this is the linking reaction. So what happens in the linking reaction? So the cycle begins with 2 carbon unit. Why? Pyruvate, which is formed in the end, loses 1 carbon dioxide and it becomes acetyl. Acetyl, and it is attached with coenzyme A. So hence we call it as acetyl CoA. So this reacts with the 4 carbon molecule. Which is that 4 carbon molecule that it reacts with? OAA, oxaloacetic acid. You have learned this. OAA is a 4 carbon. So 4 carbon and acetyl CoA 2 carbon. So now you will get, so the first molecule that's produced is citric acid or citrate. So hence this cycle is also called as citric acid cycle. So it's also called as Krebs cycle, and it is named after the discoverer, that is Sir Hans Krebs. Okay, so hence it's called as Krebs cycle. It's called as citric acid cycle, another name. Now what happens in every step? This also has 9 important steps. So what happens in each step? Can you please explain? So yeah, we did this also. You can look into your book also, that's fine. Krebs cycle, come on, tell me, where is this Krebs cycle happening?
Mitochondria.
Mitochondria. Where in mitochondria? Mitochondria has cristae, matrix, outer membrane, perimitochondrial space. So where exactly it's happening? Location.
The matrix.
In the matrix. So in the matrix, within the mitochondria, in the center of the mitochondria, the granular matrix, where all the enzymes that's required for this Krebs cycle is present. So there. So we discussed this. So Krebs cycle has, so this is the cycle. So this is the same cycle given in your textbook also. So now stepwise, so let's take up the stepwise description. I think, yeah, stepwise, what all the, how it happens. Are we learning SLP also here? So till now, how many SLPs we learned? Substrate level phosphorylation. Directly substrate, it will be generated. It's not a directly, somebody gives you rupees, you can go to the shop and, so if somebody gives you the in the form of dollar or in the form of in India, so in the local place, so you have to go for the conversion of it. So those are all energy precursor. So if you can directly spend it, so then it is the energy currency. So energy currency is ATP in our body. So precursor are so many. So all the vitamin derivative coenzymes are considered as precursors. Okay. So here NADP is also a precursor. Okay. GDP is also a precursor. Anything which is derived from the vitamin derivative coenzymes. Okay. So these are all precursors. So now, till now, how many NADP utilized or formed, and how many ATP are till now? How many? The energetics, how many we have got? See here.
NADH and 2 ATP.
2 ATP, 2 NADH. So we have got. So now we are learning continuation of the aerobic where pyruvate is converted to acetyl CoA 2 carbon compound and it enters into the matrix of the, so it passes through the porous outer membrane and enters into the matrix. So in the matrix, this acetyl-CoA, okay, combines with oxaloacetic acid to form 6-carbon compound. The first is compound, stable compound, that is citric acid or citrate. Hence, this cycle is also called as citric acid cycle. So here coenzyme is released. Okay, so now it's converted to, so we discussed that. So what is the next step?
Isomerization.
Very good. So isomerization takes place. So citrate to isocitrate. So aconitase is the enzyme. So the enzyme is given in this light blue here. So isocitrate. So isocitrate loses what? What isocitrate? Loses 1 carbon dioxide and it becomes 5-carbon compound to form alpha ketoglutarate. So hence it is a loses. So here isocitrate, the major enzyme, so that is NAD, is converted to NADH. So this is one important step. Okay. So this is an oxidative decarboxylation. What is the meaning of oxidative decarboxylation? Why do you call it oxidative decarboxylation? Name any step in the Krebs cycle where oxidative decarboxylation takes place. What is the meaning of it? What do you mean by oxidative decarboxylation? Why do you call it so?
Is it oxidative because the NAD plus is getting oxidized?
Oxidative, why? Photophosphorylation, oxidative phosphorylation. So that means what? In the presence of oxygen. So here, in the presence of? I want everyone participate, give some your inputs. So it's important, children. Why oxidative? By losing hydrogen. It is an oxidation. My voice is breaking now.
It's fine now.
Yeah, so this is an oxidative decarboxylation. So this oxidative decarboxylation, that is oxidation also happens, and that is carbon dioxide is also lost. So keep counting the oxidative decarboxylation places. So how many carbon dioxide? This is the how many number of carbon dioxide where the first carbon dioxide is lost.
When acetyl coenzyme
is formed, pyruvate to acetyl coenzyme. So keep counting the number of carbon dioxide, number of NAD formed. So every time. So let me take a different colors and color it so that. Minimize this toolbar. I don't know. Okay. So this is one of the best pictures. I got it from some book. So I'm using it. This is very clearly given. The structural formula of all the substances also given here. So it's not required for you to learn, just I'm telling you. So this is, first is the enzyme that helps in the formation of citric acid. So the enzyme, just a minute, the enzyme that helps in the formation of citric acid is isocitric acid synthase. Citric synthase. Remember, this is one of the NEET question, very important. So next is, yeah. So here carbon dioxide is lost. So now pyruvic acid becomes acetyl coenzyme A. Acetyl coenzyme A is 2 carbon, mixes with oxygen, that is now it becomes 5 carbon. 5 carbon is formed because of the loss of carbon dioxide. So the carbon dioxide lost here is the second carbon dioxide. Keep counting. So NAD formed here. So remember this. So I color that with a different shade. So that 2-3 shades if I use, so you will remember. So this is NAD. Okay. So now what is happening next? So alpha ketoglutaric acid is a 5 carbon compound. So please remember that's a 5 carbon. So now this is 5 carbon compound is formed. So now what is next? So here again one carbon dioxide is lost. Keep seeing. So again one carbon dioxide is lost, and now it becomes, this is a very important step. So alpha ketoglutaric dehydrogenase, and whenever carbon dioxide is lost, you can see the pattern. So where dehydrogenase is happening. So this is an oxidative dehydrogenase where NAD is converted to NADH, that is the energy precursor. Reduction of NAD is an energy precursor. So here coenzyme is attached. So it forms 4 carbon compound. So this 4 carbon compound that's formed is succinyl coenzyme A. Succinyl coenzyme A loses coenzyme. When it loses coenzyme, there is a surface, that is SLP takes place. So what is SLP? What is SLP? Substrate level phosphorylation. So you can see here substrate level phosphorylation. So this is the SLP 3 or 4.
4.
Yeah. SLP. This is one SLP, substrate level phosphorylation. Number the substrate level phosphorylation. So now what happens? Succinyl CoA is converted to succinic acid. So CoA is lost and ATP is released. Phosphorylation takes place. So succinic acid is reduced to fumarate. So reduction takes place. So here very, very important, succinic acid. So here succinic dehydrogenase. So keep in mind, succinic dehydrogenase is towards the perimitochondrial membrane. This enzyme is towards the perimitochondrial enzyme membrane, and it forms conversion of FAD to FADH2 takes place. So conversion of FAD to FADH2 takes place, and this is also energy precursor. What is the full form of FAD? What's the full form of FAD? Anybody? NAD is nicotinamide. What is NAD? Anybody?
Flavine.
Nicotinamide adenine dinucleotide. Now, what is FAD?
Flavine.
Flavin, vitamin A, derivative, riboflavin. Yeah, flavin.
Adenine dinucleotide.
Sorry?
Flavine, adenine
dinucleotide. So these are all energy precursors. Okay. So this happens with the help of succinic dehydrogenase, where succinic acid is converted to fumaric acid, and fumaric acid. So now succinic acid onwards, it's a 4 carbon compound. So now water is hydrolysis, that is water is used to convert this fumaric acid to malic acid and fumarase. Malic acid, one of the intermediate that's formed in photosynthesis also, you have learned that in C4 pathway. So now malic acid is converted to another oxaloacetic acid, and here also NAD is utilized. So NAD, here also NADH is formed. So how many places we have NAD? So now you can see 3 places NAD and 1 place FAD, and then 1 ATP is formed. That is substrate level phosphorylation is formed here. So now, what is the balance sheet? So let's go to the balance sheet. See, second, the first oxidative decarboxylation, second oxidative decarboxylation, and here, so where all carbon dioxide is released? Carbon dioxide is released in 2 places. So it is in where all carbon dioxide is released in 2 places, and in pyruvate 1 place. So you can see in 3 places this carbon dioxide is released. Okay, so this complete the Krebs cycle. Okay, oxidation of succinic acid produces FADH2. So every day, so hence it is called as oxidation. And along with oxidation, decarboxylation occurs. Oxidative decarboxylation, that's called as. See, the third NADH is produced here in this cycle, and it comes to the regeneration of oxidative OAA, that is oxaloacetic acid. So now come on, answer all these questions. So then all about Krebs cycle is, to some extent, your knowledge is clear. So now before going to those questions, I would like to give you this balance sheet. So fats also get converted to acetyl-CoA. So that conversion of fat to acetyl-CoA is called as beta oxidation. So the process of conversion of fat to acetyl-CoA is called as beta oxidation. So they undergo beta oxidation reaction for the conversion of. So if at all there are so much of fat in your body, so the fat enters into the Krebs cycle. Even they can become the substrate for the formation of, right? Now, what's the significance? How many NAD produced so far?
8.
I want to answer from Akash. Throughout the Krebs cycle, how many NAD formed? So this is one of the detailed Krebs cycle. I got it in some foreign book, that is foreign author book. So I took it from that. So how many NAD formed so far? Akash, it's so clearly given there only. NAD is formed here. NAD is formed here. NAD is formed here. Yeah?
3 plates.
Yeah. So the third NAD, the second NAD, which is given there. So NAD formed there. So 3 into 2. Why do we write 2?
Because there's 2 pyruvate molecules.
So every time 1 glucose molecule give rise to 2 pyruvate, and every time 2, 2, 2, 2, we are writing, that is 6. So now 2 FAD, because once in one place. So when the FAD is formed? When, in which place FAD is formed? Oxidation of succinic acid. When succinic acid, okay, get converted to fumaric acid. Fumarate or succinate, one and the same. Succinic acid or succinate, one and the same, sorry. Succinic acid to fumaric acid or succinate to fumarate. So their oxidation takes place. So then, because of the oxidation, so NAD is reduced. Okay, so NAD is reduced or FAD is reduced here. FAD is reduced to FADH2. So there, that is a very important point. So there to FAD. and 2 ATP. So that's a substrate level phosphorylation. So substrate level phosphorylation takes place when the succinyl-CoA loses coenzyme A and becomes succinic acid. Succinyl-CoA loses succinic CoA and becomes succinic acid. So this is the balance sheet of, that is 6 NADH, 2 FADH2 and 2 ATP. So what's the significance of this Krebs cycle? So it's a common pathway of oxidation, breakdown of, so it's a common pathway, Krebs cycle. So oxidative breakdown of carbohydrate, fatty acids, and amino acids, everything. So anything can become the substrate level, substrate for the respiration. But everything can get converted to acetyl-CoA before entering. Okay. So now this is an amphibolic, though there is a breakdown of the glucose to pyruvic acid. Pyruvic acid is a further breakdown to different intermediate products. So this is I'm sorry, something happened and it got disconnected. Can you hear me?
Yes, ma'am.
Can you hear me?
Yes, ma'am. We can hear you.
Okay. So can you see the screen?
Yes, ma'am.
Okay. So I was telling you that though this is a catabolic biochemical pathways, so many of the intermediate products are used up further for the formation of different substances that for the plant body or any other organ, even in your body. This is one, we are not talking about the cellular oxidation with respect to plants alone here. So this cellular oxidation takes place in any aerobic organism. Okay, so in the cell, this is how the pathway occurs. So this is a common part of oxidative breakdown of carbohydrates, proteins, and amino acids. And here, so every time we are getting an important substances. So what's that importance of these substances? Let's take a look here. So alpha ketogluteric acid produces glutamate. So that is glutamic acid. That's a very important amino acids that's required for the synthesis of fatty acids, cuteins. So with respect to plants, so it's used to produce cutin. So we have learned that cutin in biomolecules, that cutin, that's cuticle, waxy coating, suberin or wax derived substances that we get from plant. So this produces that butene, okay? And this produces aromatic compounds, isoprenoids, okay? So all these are required for the formation of carotenoids, terpenes, gibberellins. Gibberellins is a phytohormone, okay? So all these, so hence it's used to build up some other substances in the body. Point number 2, succinyl CoA is the major compound, so that's required for the formation of the cytochromes. So cytochromes are the carriers, electron carriers. So through Krebs cycle, so that is, we are building many other substances. Hence, though it is involved in catabolic reaction, the intermediates are used for the building up of the different biomolecules that's required in the living system. So therefore, Krebs cycle is not just a catabolic processes, not just a catabolic cycle, it's also an amphibolic. So amphibolic means what? So both catabolic and anabolic. So we can consider the Krebs cycle as amphibolic pathway. So this is an important subjective question they would ask. So you need to cite some of these significances of and intermediate products, how they are used up for the different building up processes. Now, let's go to the further concept today. Still now we did it. Akash, this is for you. We did the recap fast. It's a fast recap. If you go through the video and the notes, which I'm sending the same notes soon after the class, so you would understand much better. Right?
Yeah.
Yeah. So now, when we say that energy is produced utilizing glucose, so what's that glucose means? So why organic substances? It's not alone glucose, amino acids or fatty acids. So it's a bond carbon and hydrogen that is used. The covalent bond that's in the organic compounds. So these produce electrons. So these electrons. So this is the covalent bond that is there in the organic compounds. Okay. So oxygen is required here in the aerobic respiration for the formation of water. And instead of oxygen, so this hydrogen is that is reduction of this oxygen releases water here and reduction of hydrogen, so that you get, so that's why we say that fermentation is an incomplete oxidation. Whereas aerobic respiration, there is complete oxidation. So what happens here, there is a change in the free energy when the bond breaks. So this change in the free energy releases enormous amount of energy, and it's about 686 kilocalories. Okay, so the bond that's present in the covalent bond that's present in the organic compounds, when they break, so it forms a change in the free energy, and that releases the energy which is utilized to create ATP. So here, every time hydrogen are removed, so like how we understood in chemiosmosis, in photosynthesis, hydrogen proton gradient is formed. So this forms the redox potential. So this proton gradient is very much required for generation of ATP. So that's how the phosphorylation took place there. And that's how the phosphorylation takes place here also. But the proton gradient that's formed here is enormous compared to that. So here, when every time hydrogen is removed during the bond, when the bond breaks, so it's transferred. That is, when hydrogen is removed, that's a proton is removed, even the electrons are removed. So the transfer of this electron, okay, so takes place from one enzyme, coenzyme to another, or the electron carriers, which we call as electron transport chain. So this electron transport chain only we are learning now. So in phosphorylation also we learned the same thing in photosynthesis, cyclic phosphorylation and non-cyclic phosphorylation, because of which proton gradient is formed. And this proton gradient helps in the formation of that is F1, F0 particles help in the formation of ATP. You have learned that. The same thing happens. Chemiosmosis here also takes place. So this electron transport chain. So where exactly this happens? So this happens in the inner space between the two membranes. So there it happens. So every time, so NADH and FADH2 releases 2 electrons, okay, formed during the first 3 stage of the aerobic respiration. So how it happens, this is the same picture given in your textbook also. This is the same picture given in your textbook. Is it there?
Yes, ma'am.
Yeah, I want you people open your textbook. Open your textbook. Is my voice breaking still?
Oh, ma'am.
Give me water. Pani de do. Yeah. Is this picture given in your textbook? I mean, something like that only it is given, right?
Yes, ma'am.
Yeah. So did you understand till now, all of you, before moving ahead?
Yes, ma'am.
I'm not getting that yes, ma'am, from other students. Bihan, why the electron has to be transported? Where this electron come from? Okay, answer. If you know all this, these are the questions children always wonder. Where this electron come from? Yes.
The NADH.
Sorry?
NADH molecule.
See, when bond breaks in the organic compounds, so hydrogen is removed from every time. So, okay, so then it is transferred. So when hydrogen is removed, so along with that, even the electrons so are removed. So that's from every time it is removed. So these electrons, okay, and so that is transfer of these 2 electrons and 1 proton. So when it is moving from NAD to NAD, so that's how it is converted. NAD is converted to NADH because of the movement of protons. So then movement of electron also takes place. But we are learning this separately here. All these happen simultaneously. Are you understanding? We are learning step after step, but it is not that it will wait for that to happen and then this happens. It's not like that. Simultaneously, it's like a factory. Simultaneously, these things will be happening. The moment protons go to NAD and NAD is reduced to NADH to conserve energy. Similarly, at the same time, electrons are also moved for the carriers, electron carriers. Are you understanding? So where this happens, so you can see this is the matrix, this is the inner mitochondrial membrane. So what do you call that inner mitochondrial membrane? What name is given to that? What name do we give it to inner mitochondrial membrane? Array guys, this is your 9th standard. What name is given to that? Anybody? Christe, have you heard this name?
But Christy are the enfoldings, right?
Sorry?
Christy are the foldings.
Inner mitochondrial membrane is folded, thrown into folding. And what do you call it as? Cristae. So cristae are the foldings. So they are nothing but the second. So mitochondria is a double membrane cell organelle, just like plastid. Right? And it is also semi-autonomous. Colliker and Benda discovered it. So why is it semi-autonomous? Because it has its own DNA, it has its own ribosomes. So the outer membrane is very porous. So it allows anything to pass. So it's very porous. So inner membrane is folded to give the large surface area. For what? So the large surface area too for the catalytic site. So it holds that on the inner membrane of mitochondria when it is folded like this. So it has a racket-shaped structures. So what are these racket-shaped structures? F0 and F1 particles, coupling factors, mitochondrial coupling factors. So F0 is embedded in the membrane and F1 act as ATP synthase only during the formation of ATP or during phosphorylation. So that's what happens. But before the phosphorylation, how the electrons will move from one complex to another complex, that's what we need to know here. Just like chemiosmosis. Are you understanding?
Yes, ma'am.
Yeah. So now there are 4 important complexes that's involved in electron transport here. Which are those 4 complexes that's given in your book? Complex number 1, dehydrogenase, NADH dehydrogenase. Complex number 2, succinic dehydrogenase. Complex number 3, cytochrome BCF, that is reductase. And complex number 4, cytochrome C oxidase. Is that given in your textbook?
Yes, ma'am.
There are 4 complexes that's there in the inner mitochondrial space. Is it there?
Yes, ma'am.
Yeah. So what is this system? This system is called electron transport system or electron transport chain, or also you can call it as oxidative phosphorylation because phosphorylation takes place. So if they ask you the question, what is oxidative phosphorylation? What answer are you going to write? Are you all getting to see the screen?
Yes, ma'am.
Yes, ma'am.
So what is that you're getting to see on the screen?
The diagram.
Sorry? The picture?
Diagram. Yes, ma'am.
Yeah. So what is electron transport system? If they ask you this question in general, this can be asked for photosynthesis also, this can be asked for this also. What answer are you going to write? What is electron transport system?
The system involved.
Metabolic path. Yeah. The system that's involved. Perfect. Yeah, tell me, Purvi.
System involved in the
Good try.
transport of electron for the synthesis of ATP at the end.
Yeah, the system that's involved in the passing of electrons from one complex to another complex is electron transport system, and that's what here this complex is present in the inner mitochondrial membrane. So this consists of a series of electron transport complexes. These are coenzymes. Okay, so like NAD and flavin adenine FAD, ubiquinone, cytochrome B, cytochrome C, cytochrome A, cytochrome A3, different cytochromes we discussed. So now here there are majorly 4 complexes and 2 mobile carriers. You can mark it. 2 complexes and 2 carriers. So let me write that here. So there are, is it given somewhere here? Yeah, let me write here. So there are majorly 4 complexes and 2 mobile carriers. And 2 mobile carriers. Right?
Yes, ma'am.
So which are those complexes? You can see on the picture. Which are those complexes? Let me take the color to highlight it. Okay. Which color shall we take to highlight? So whichever is not there, we can take that color. Yes. So this is complex 1. It's not shown, no. So let me take this. This is complex 1, you can see. So what is this complex 1 made of? Come on, answer. Yeah, can you see the complex 1? What this complex 1 is made of?
Ma'am, I think you stopped sharing.
Sorry? Yeah, again?
Ma'am, we can't see your screen.
Stop, is it? No, I'm getting it shown.
Now we can see.
It is coming and it is shown only. Guys, please tell me what this complex 1 made of. I have highlighted this complex here. What's the name of the complex?
NADH dehydrogenase.
Yeah. So, flavoprotein. So, what is complex 2? Complex 2 is here. First, you understand the complex 1, complex 2, all these. So, what is this complex 2 made of?
Succinate dehydrogenase.
Here, complex 2. Yeah. Give me the name. Give me the name. Read it, guys. Read it. Complex 2 is made of.
Succinate.
Complex 2 is made of. Complex 2 is made of.
Succinate dehydrogenase.
Read the succinic acid. Succinate dehydrogenase. So I told you to remember succinate dehydrogenase is towards the mitochondrial membrane. So it's present at the mitochondrial membrane. Though it is used in the Krebs cycle, it is one of the complexes for the carrying electron. Okay, so one important point about succinic dehydrogenase is, so there is a competitive inhibitor. So what's the competitive inhibitor for this enzyme? Succinic acid, malonic acid, please write, this is your neat point. This is your neat point. That's why I have written here. So malonic acid is the acid that imitates succinic acid, and hence it's a competitive inhibitor. So succinic dehydrogenase helps in the conversion of succinic acid to fumaric acid. So we came across that in the Krebs cycle. So suppose if malonic acid is present in the plant, succinic dehydrogenase would bind to that succinic malonic acid instead of succinic acid, and thus will stop the respiration only. Respiration would not continue. So because this is a competitive inhibitor, malonic acid, please write down that, mark that as your important need point. So now the complex 3, which is a complex 3?
Cytochrome.
Complex 3 is
Cytochrome.
cytochrome what? Cytochrome B and C1. So here it is given cytochrome B and C1. Here, this is complex 3, cytochrome B, C1, right? So what's the speciality about this particular complex? So how is it different? So between this complex 2 and complex 3, so what do you find between complex 2 and complex 3? What's that you people can find? So you can see this. Oh, it's fully dark. What do you see? If you have learned already this, so you can see this.
Ubiquinone.
Ubiquinone. Exactly. So what is the we discussed all about ubiquinone. What is ubiquinone? It's used in the quinone cycle. So ubiquinone is involved in quinone cycle. So quinone cycle's function is what? Mobile carrier. Ubiquinone is a mobile carrier. It's involved in quinone cycle. So that means it will get reduced to ubiquinol if at all it has to carry the electron, right? So while transferring electron, ubiquinone, so it get reduced. So once it get reduced, after passing the electron, what it does? So it pumps the proton into the inner membrane space, perimembrane space, perimitochondrial space. So therefore, the pumping of electrons, so that is hydrogen is happening, the proton or hydrogen. So we call it proton. So proton gradient is forming in the inner mitochondrial space. So let us understand that completely. So which is the complex 4? Come on, complex 4, which one? Complex 4 is given here. So you should know all the complexes by heart in your mind. So then only you can simply write electron transport system. This is your 5-mark question, and lot of questions can come from this. So which is a complex 4?
Cytochrome C oxidase.
Cytochrome C or A?
E.
Cytochrome C is given. So that is it. In your textbook, is it given C or A?
E.
What's given in your textbook? NCERT?
It's given C.
Okay, cytochrome C is a copper. Okay, B and CF is a non-heme. So here it is heme. So this is copper. C oxidase is a copper based. You can write down. That's a again. So neat question. So cytochrome T is a copper-based. Copper-based cytochrome. Okay, so second is C-oxide. So second is, where else you find cytochrome? 3. Complex 3 is B and C. Okay, so this is this one. So now electrons from NADH that's produced in the matrix. When the electrons are produced? When it reduce NAD to NADH, so electrons are transferred in Crick acid cycle, Krebs cycle. Okay, so the Krebs cycle. So who carries every time? Who carries this electrons?
Ubiquinone.
Ubiquinone. So what ubiquinone is doing? So whenever there is a reduction, so you can see here, reduction of NAD to NADH. So hydrogen is pumped into this, right? So this particular complex, that is NADH dehydrogenase, the complex 1, is a ubiquinone oxidoreductase. It's also called as NADH ubiquinone oxidoreductase. So it's an iron sulfur complex. So please write down. So here I have marked. So this is an iron sulfur complex. Look here. Okay, so this ubiquinone, while carrying the electron, it picks up the hydrogen proton from the matrix. So, and it is reduced to ubiquinol. So ubiquinone is reduced to ubiquinol. So this is ubiquinol. So after passing the electron to the complex 2, so see here, ubiquinol comes from here, it pumps the 4 hydrogen and comes and gives the electron. So when it passes to succinate dehydrogenase for the conversion of the succinic acid to fumaric acid, right? So now the cytochrome comes in place. So this cytochrome, okay, so picks up the, see here, so this cytochrome picks up the electron and is passed on to the next. So where the final electron acceptor is, who is the final electron acceptor? This is an important question in your this one also. Who is the final electron acceptor?
Oxygen.
Oxygen. Look here, oxygen is reduced. So that means its final electron acceptor, which accepts the electron to form water. So every time when electron is moving from one place to another place, the protons are pumped into the perimitochondrial space, thus forming the electrochemical gradient. So proton gradient is formed. Okay, so this proton gradient results. So finally, so in the there are oxysomes or F0, F1 particles in the Fernandez-Morens particles. They are also called as. So they are present in the membrane of this cristae, where this is the F0 act as an, this is F0 act as an embedded proton channel, same way. And this is the catalytic site which act as ATP synthase. So which results in the formation of ATP, and hydrogen is pumped from this perimitochondrial space or inner mitochondrial space into the matrix back. So when it is movement, the movement of this proton helps in the release of ATP. So this is called as oxidative phosphorylation, as there is in the presence of oxygen. Right? So how this oxidative phosphorylation takes place? With the help of energy that is formed during oxidation of reduced coenzymes. So that means coenzymes get reduced. So hence, this is also a chemiosmosis according to Peter Michel. So this is the perimitochondrial space. So for every 3 hydrogen protons that's pumped, 1 ATP is released, and this is F0, which is embedded in the channel, and this is F1, which is acting as ATP synthase. Is that clear? So what is the complete balance sheet here then? This is a complete balance sheet, 3-mark question. So for each balance sheet, they will assign 1 marker. So the net ATP that's formed during glycolysis is 2. NAD is formed is 2. So every 1 NADH produce 2 ATP. So therefore total 4 ATP. So in the link reaction, there are 2 NAD, right? NAD and H plus. So therefore there 6 ATP, so total here. So Krebs cycle, how many? 6 NAD, right? NAD and H plus. So remember, this is only NADH, and here NADH and H plus. So therefore 3, 6 3s are 80. Now FAD produces, so 2 FAD, 2 2s are 4, and 2 ATP in the SLP. So that is here, that is 2. Substrate level phosphorylation. When the coenzyme succinyl-CoA becomes succinate. So therefore, there are the net ATP that's formed is 36. So the gross ATP that's formed is, in the equation we put 38. So gross ATP is 38, where 2 is utilized in the investment phase in the glycolysis, and hence net is 36 ATP. Right? So I want Akash read electron transport system from your textbook once, every bit. And then we will move ahead with this. All about electron transport system, I have taken the questions here. So all about Krebs cycle, I have taken the questions. So if we do these questions, so your concept become much better. Quickly. Akash, please read it from your textbook.
Okay.
Electron transport chain.
Electron transport system. Right?
Yeah.
The following steps in the respiratory process are to release and utilize the energy stored in NADH+, H+, and FADH2. This is accomplished when they are oxidized through the electron transport system, and the electrons are passed on to oxygen, resulting in the formation of water. The metabolic pathway through which the electron passes from one carrier to another is called the electron transport system, and it is present in the inner mitochondrial membrane. Electrons from NADH produced in the mitochondrial matrix during citric acid cycle are oxidized by NADH dehydrogenase complex 1, and the electrons are then transferred to ubiquinone located within the inner membrane. Ubiquinone also receives reducing equivalents via FADH2 complex 2 that is generated during oxidation of succinate in the citric acid cycle. The reduced ubiquinol is then oxidized with the transfer of electrons to cytochrome C via cytochrome BC1 complex 3. Cytochrome C is a small protein attached to the outer surface of the inner membrane and acts as a mobile carrier for transfer of electrons between complex 3 and 4. Complex 4 refers to cytochrome C oxidase complex containing cytochromes A and A3 and 2 copper centers. When the electrons pass from one carrier to another via complex 1 to 4 in the electron transport chain, they are coupled to ATP synthase complex 5 with the production of ATP from ADP and inorganic phosphate. The number of ATP molecules synthesized depends on the nature of the electron donor. Oxidation of one molecule of NADH gives rise to 3 molecules of ATP, while that of one molecule of FADH2 produces 2 molecules of ATP. Although the aerobic process of respiration takes place only in the presence of oxygen, The role of oxygen is limited to the terminal stage of the process. Yet the presence of oxygen is vital, since it drives the whole process by removing hydrogen from the system. Oxygen acts as the final hydrogen acceptor. Unlike photosphorylation, where it is the light energy that is utilized for the production of proton gradient required for phosphorylation, in respiration, it is the energy of oxidation reduction utilized for the same process. It is for this reason that the process is called oxidative phosphorylation. You have already studied in the mechanism of ATP synthesis as mentioned by
chemiosmotic process. Yeah, chemiosmosis means Peter Michel. The chemiosmosis is the same process. So whether it is plastid or whether it is mitochondria, chemiosmosis is the pumping of this, that is bringing down the proton gradient, formation of the proton gradient, where the movement of the protons from the higher concentration to the lower concentration gives the energy. So where coupling factors, F0 and F1 particles act as the coupling factors to create that conversion of or bring in phosphorylation. Okay, F0 and F1 particles. So now, Bihan, give me the complete differences between glycolysis and Krebs cycle. Then we will go ahead with these questions, neat questions. Because there should be complete understanding of this concept. So then once you revise, so glancing becomes easy. Vihan, give me the complete picture about the differences of Krebs cycle and glycolysis.
Ma'am, in glycolysis, the 6-carbon molecule is broken down into a 3-carbon molecule.
Your voice is breaking. Why? Yeah, give me the answer now. Now you speak. Now you speak.
In glycolysis, the 6-carbon molecule is broken down to a 3-carbon molecule.
No, very first and the foremost, what is that you should tell? Glycolysis, I have not written this. So this is an important, jot down the points. Glycolysis occurs in cytoplasm and Krebs cycle occurs in the matrix of mitochondria. That's a very first and the foremost difference. So what's next? Glycolysis is mandatory, compulsorily occurs, the first process in both irrespective of aerobic or anaerobic, whereas Krebs cycle occurs only in aerobic respiration. Glycolysis is not a cycle, it's a linear pathway, whereas Krebs is a cyclic pathway. Citric acid cycle is a cyclic pathway, right? So these are all very simple differences which you should know. So what happens in glycolysis? What's the end product in glycolysis?
Pyruvate.
Pyruvate. What's the end here in Krebs cycle? So pyruvate is oxidized. Glucose is oxidized to pyruvate. So it's not a complete oxidation. So here, acetyl coenzyme is completely oxidized. So here, consumption. Now comes the last comes the energetics, that is the how much ATP all these utilized, and then release of CO2. So CO2 is not at all produced in glycolysis. Please remember, this is one important point. CO2 is never released in glycolysis, whereas in Krebs cycle CO2 is released. Another important point, So all the enzymes that's required for glycolysis is present in cytosol, cytoplasm only. Cytosol, we should say, if it is only liquid part. So the enzymes that's required for Krebs cycle is located both in the matrix and inner mitochondrial membrane, like succinic dehydrogenase, just now we discussed. It's one of the complexes in the electron transport. So succinic dehydrogenase helps in the formation of succinic acid to fumaric acid. Right, very important complex. So this complex is not in the Krebs, in the matrix, but it's involved in Krebs cycle. It's present on the membrane. One example. So lastly, the energetics. So 2 ATPs are consumed. Here, no ATP is consumed in Krebs cycle. So here, 2 ATPs are released and 2 NADH are released. Here, 6 NADH are released, 2 FAD are released, 2 ATPs are released. So when they are released, that's what is the need point. Please remember. So for subjective, you can remember only this much. For need point, you need to remember complete information. So when they are released, how many carbon dioxide released, in which phase it is released, all these you need to remember for your need. So for that, you need to practice the cycle. That's it. So now, how many of you have noticed or heard or seen in the movies that so these terrorists consume cyanide if they get caught? That's what we have seen, no? So why do they do not survive? So that the moment they eat up cyanide or inhalation of cyanide gas or cyanide that substance, so within no time they die. Why? The suffocation happens and die. Why? Why? Any idea? Anybody? What could be the reason? This is connected to the Krebs cycle. That's why I mean electron transport and Krebs cycle. Why? Which is the final electron acceptor?
Oxygen.
Oxygen. So oxygen. So for that oxygen, who transports electron?
The ubiquinone.
Ubiquinone carries it to the mobile. It's a mobile carrier. It carries it to the coenzyme. Which coenzyme transports the electron to the final acceptor? Final electron acceptor. Which coenzyme transfers it? Look into your book. Oh, look into the picture I'm showing. Oh, I stopped 1 second. Electron transport. Who carries it? Who gives it? Can you see here? Cytochrome. See here, ubiquinone brings it and gives it. Cytochrome A, and here A. Cytochrome A3. Remember that. Now, what happens if they cyanide? So from cytochrome A3, so this cyanide binds to cytochrome A3 and stops the movement of electron to the final acceptor, that is oxygen. So when oxygen is still free, the reduction of oxygen only would result in the formation of water and the proton gradient to flow. So when the oxygen is still free, so then the proton gradient chemiosmosis would not occur, phosphorylation would not occur, formation of water would not occur, and the person immediately dies. So that is why cyanide is the deadly poisonous. Is it only cyanide? So 2,4-DNP, that is 2,4-dinitrophenol is also a, so you can write these points. Cyanide, 2,4-D, 2,4-dinitrophenol. So what are these? These are respiratory chain inhibitors. Dinitrophenol. So it is dinitrophenol. They the movement of electron instead of coming from ubiquinone to, so you can see ubiquinone should bring it to the next. So that is the complex number 3, 2 to complex number 3, cytochrome BC. So from there it has to move to the cytochrome A. But ubiquinone, this dinitrophenol directs the electron transport from quinone to another quinone Q2. So this binds to cytochrome A3. So the electron is not carried on to the, so A3 to oxygen it should go, it doesn't happen. And this directs the movement of electron transfer from ubiquinone to ubiquinone 2. So what happens? Instead of going to the cytochrome, it goes to Q2. So what happens if they go to Q2? So the further electron transport would not take place, phosphorylation would not happen, and oxygen will be free. Same thing. The result is same. Okay. So there is another antimycin A. All these are substances. So this is antimycin A is a drug. Okay. So stop the movement of electrons to cytochrome C and C. Cytochrome B2, so you can see it's going. You can see the arrow mark here. The arrow mark cytochrome B complex to the cytochrome C. So it prevents the movement of cytochrome B to C1. Cytochrome prevents electrons movement from cytochrome B to cytochrome C1. So these are some of the respiratory chain inhibitors. Okay. Is this much clear? So now I want all of you give me the yes before going to this Krebs cycle questions. Yeah. Go ahead. So let's give me Vihan, Vihan Poli. Read. Akash, read the question. Read, Akash. Am I audible?
Yes, ma'am.
Akash, please read the question.
The number of substrate level phosphorylation in one term of citric acid cycle is one B.
When it happens? The number of substrate level phosphorylation in one turn of citric acid cycle is when it happens. Go to your textbook and see. Substrate level phosphorylation, I told you, no? When that happens here, guys, when the succinyl-CoA loses, so when the succinyl-CoA loses coenzyme A, it forms GTP, which is nothing but ATP only. Okay? So when hydrogen is added, so it coenzyme A, it loses coenzyme A to become succinic acid. Succinyl-CoA to succinic acid. So is that particular cycle is given in your textbook? Look into your textbook, is it given?
No, ma'am.
Add it. Succinic acid, before becoming succinic acid gets the enzyme A. Succinyl-CoA to succinic acid. Then GTP is synthesized, which is phosphorylation. Show you the yeah. Look here, this one. So this is a substrate level phosphorylation. I have written also here substrate level phosphorylation. Formation of ATP is a substrate level phosphorylation. Only one happens here. Okay. So alpha ketogluteric acid becomes succinyl-CoA. It doesn't become succinic acid directly. So in your textbook, they have given succinic acid. So it becomes succinyl-CoA. It loses carbon dioxide. Hence, it is a oxidative decarboxylation. The second oxidative decarboxylation. So very important. So this cycle is the very appropriate cycle. So please follow this cycle and learn it through from this picture. Second one, Vihan. Quickly, go ahead, go ahead. Don't waste time. Vihan, read the question. Vihan, please read the question.
Where is the respiratory electron transport system located in plants?
Where is it located?
Ma'am, the
This is
inner
so easy. What's the answer?
Ma'am, the inner mitochondrial membrane.
Inner mitochondrial membrane. That's right. So where the proton gradient is formed? Where the proton gradient is formed? Where the proton gradient is formed, guys? Inner mitochondrial space, perimitochondrial space. In between the two membrane, the space is called perimitochondrial space. In that, proton gradient is formed. So X1, that is C0, C1, that is F0, F1, sorry. F0, F1 particles are called coupling factors. Please remember. So here it is from succinyl-CoA to succinic acid, where GDP to GTP formation takes place. Okay, so now which of these statements? Next, Purvi.
Enzymes of TCA cycle are present in mitochondrial matrix. Glycolysis occurs in cytosol. Glycolysis operates as long as it is supplied with NAD that can pick up hydrogen atoms. Oxidative phosphorylation takes place in outer mitochondrial space.
Your voice breaks.
Oh.
Your voice is breaking terribly. Can you read it?
Is it fine now?
Yes, it's better. Yes.
Ma'am, the answer is D, I think.
Why?
Phosphorylation takes place. It takes place in inner mitochondrial membrane.
Enzymes of TA. TCA?
Krebs cycle.
What is the full form of TCA? It's called citric acid cycle, Krebs cycle, TCA cycle. What is TCA?
Tricarboxylic acid cycle.
Tricarboxylic acid. Which are the tricarboxylic acids that are formed in the beginning? Which are those? Citric acid, isocitric acid, alpha ketogluteric acid are all tricarboxylic acids. You can see the structural. I have given, this is the best picture I told you as before also. So you can see here, sorry. See here, carboxylic acid group. The functional group is carboxylic acid, and see here. Are you all seeing? I'm highlighting. Yeah, so all these three are three carboxylic acid which are formed in a series. Hence, it's called as TCA cycle, tricarboxylic acid cycle. What are the names given to this cycle? So it is Krebs cycle, named after Sir Hans Krebs. Krebs cycle. It's called as TCA, tricarboxylic acid. So this is one, and this is one. So citric acid, isocitric acid, and alpha ketogluteric acid. So it's also called as, what's the other name? Citric acid cycle. Citric acid, because the first compound that's formed is citric acid or citrate. So therefore, these are the three names given to this cycle. Okay. So this is right. It's right. So glycol is occurs inside this. Glycol is optic as long it is supplied with NAD that can pick up hydrogen. Yes, that's right. And oxidative phosphorylation takes place in the outer mitochondrial membrane. This is the wrong point in the inner. So therefore, answer is this one. Okay, next one. Next again, the next person. Keep going. Akash.
Which statement is wrong for Krebs cycle?
Read the statement.
Oh, 1A, there is one point in the cycle where FAD plus is reduced to FADH2. B, during conversion of succinyl COA to succinic acid, a molecule of GDP is synthesized. C, the cycle starts with condensation of acetyl group with pyruvic acid to yield citric acid. D, there are 3 points in the cycle where NAD plus is reduced to NADH plus H plus. Ma'am, it's C.
Yeah, good. So what's the right answer, right statement for it? There is one point in the cycle where FAD is reduced to FADH2. Yes. So during conversion of successful molecule, GDP synthesized. Yes. The cycle starts conversion of acetyl group with pyruvic acid to yield citric acid. No. So it's what's the right? The three. Okay, there are 3 points in the cycle where NAD is reduced. Yes, that's also right. So which are those 3 points you should know? Please learn. So what's the right answer for C then?
Ma'am, it's condensation of acetyl group with pyruvic acid along with water.
No. Conversion of, what's the right statement for that?
Yeah.
The cycle starts with condensation of acetyl CoA with pyruvic acid or with what? Is it with pyruvic acid? That's a wrong thing here. It's not with pyruvic acid. To yield citric acid, condensation of the acetyl CoA takes place with what? Anybody?
Oxaloacetic acid.
Oxaloacetic acid. Acetyl CoA combines with OAA to form citric acid. So the answer is oxaloacetic acid. The next one. Who's the next one? Purvi, it's yours.
Oh. Oxidative phosphorylation is formation of ATP by transfer of phosphate group from a substrate to ADP. Oxidation of phosphate group in ATP. Addition of phosphate group to ATP. Formation of ATP by energy released from electrons removed during substrate oxidation. I think it's D.
Why D? What's the reason?
Ma'am, because after the ETC, the electrons which are removed from NADH, they travel the ETC and then at the end, they combine with oxygen to form water and energy.
So this is an easy one. Removed during. So this is an easy one. In mitochondria, protons accumulate in. This is also an easy one. Outer membrane, inner mitochondrial space, and matrix. There are only 4 spaces. Where inner mitochondrial space, I know this is an okay, next one is a good one. So who will answer? So I'm not going to, let me see who is interested to answer or have understood everything so that I'll come to know. 19th question. Good one. Someone is saying something, but I'm not able to hear.
Ma'am, isn't it succinate dehydrogenase?
Vihan, right?
No, it's Akash.
Akash, okay. Okay, Akash, come on.
It's C.
Sorry?
C.
B.
C. Succinate dehydrogenase.
Yeah. So except one, that is, see, this is also very important one. Next, 28th one. Two names refer to the same thing.
E, tricarboxylic and citric acid cycle.
What are the names given to Calvin cycle?
C3
Do
cycle.
you remember? This is where students always get confused. They jumble up, Calvin cycle, Krebs cycle. Calvin is the Melvin Calvin, Sir Hans. There it is C cycle, here it is citric cycle. So tricarboxylic acid cycle. What is the other cycle name? It's given there. Biosynthetic phase. There. Okay, so those are the names given there. So which of the following statement about cytochrome P450 is wrong? What is that P450? So what's that cytochrome P450? That's a very crucial question. Anybody? Could you figure out that cytochrome 450? Yeah, anyone? Yeah. Okay, what are cytochromes? So they are the electron transferring proteins. So they have iron porphyrin or
It's iron, I think. Oh, ma'am, is it copper?
Oh my God, I was on, kept on and talking. When I got disconnected, I'm not aware of.
Ma'am,
Can you hear me?
Yes, ma'am.
Can you hear me?
Yes, ma'am.
So what, yeah, we were talking about, did you hear when I told cytochrome P450?
Yes, ma'am.
Yeah, yeah. So what is your answer? P450, what is that? P for plant, remember this? P for plant, plant 450. Okay, they are found in some microorganisms also. So they actually, they are involved in different reaction, hydroxylation, dealkylation, right? So they are involved in different functions and therefore they are colored cytochromes, colored cytochromes. So that's your answer. I'm sorry, wrong answer. So they are colored cell. That's your wrong answer. Let me color it. They are involved in different functions. Okay. Yes. So important role in metabolism. Cytochromes means they are iron content. So iron, sulfur or copper. So I told you iron porphyrins or copper porphyrins, cytochromes. So remember that. Cytochromes are chromes are iron or copper porphyrin structure. Porphyrin means having metal in the center. Next one, which of the following is the key intermediate? I do one thing. So 1, 2, 3, 4, 5 questions. You do it. 5, 6, 7. 7 questions I have added here. End product of it. So please do that. This is an easy. So now let's go to electron transport system. So there some questions are there. These are all very crucial questions. So whatever is crucial only I have taken and added here. Okay, so now there are only 5. Answer this, then we can go further. Terminal cytochrome of respiratory chain which donates electron to oxygen is? Very nice question.
Cytochrome C.
Give the answer.
Cytochrome C.
Yeah. Cytochrome. Terminal cytochrome. Which one?
E.
No, 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 no. Terminal, my dear. Just now we discussed about cyanide. Then I told you, sir, terminal, which gives the electron finally to the oxygen for the formation of water. Who is that? Cytochrome A3. I wrote no, cytochrome A3. So cyanide binds to that. Cytochromes are found in where? Is it only in the cristae of mitochondria or, okay, here with respect to cristae, matrix, where do you find them? Cytochrome. This is also a very important question, according to me. Where do you find cytochrome?
Cristae.
So cytochromes are found in? Yes, I'm not getting any answer from boys. Where do you find cytochromes, boys?
Crystal.
Is it only in the cristae?
Yeah, mitochondria.
Yes.
Yeah.
So cytochromes are found in cristae. Next. Next. Quickly read the questions. Cytochrome is iron containing porphyrin, metalloflavoprotein, glycoprotein. What's your answer? Cytochrome is
Iron containing porphyrin.
so it's an cytochromes are porphyrin ring. Correct. So iron and or copper porphyrin. Okay, so the correct answer is metalloflavo. What's the correct answer? What do you think is the correct answer? Any metal? No. Iron or porphyrin. So here the more appropriate is iron. So this we did it. This is not. So all these are yes. Cytochrome sequence of okay. This is a very nice question. 33rd question I liked a lot. So who can give the answer? This is a very nice question. The correct sequence of electron accepted in ATP synthesis. Who can give the answer?
BCAA3.
So option
D.
very good. This is a nice, very nice question. So in the last, so aerobic respiration, oxidation of reduced coenzymes. So all those which are reduced should get oxidized. So they, how it happens? By the molecular oxygen. So ubiquinone is the, so it is BC1CA3 actually. Okay. BC1CA3. So this is how it happens. So A3 gives it to finally to A3 to oxygen for the formation of water. So how many ATP molecules? The last one maximally be generated from one. Someone read the question. Akash, read the question.
How many ATP molecules could maximally be generated from one molecule of glucose if the complete oxidation of 1 mole of glucose to CO2 and H2O yields 686 kilocalories and the useful chemical energy available in the high energy phosphate bond of 1 mole of ATP is 12 kilocalories?
What's your answer? What's your answer?
Is it 57?
Why?
Yeah, it is 57. 686.
Why 57? Why? How did you calculate?
686 by 12.
See here, one mole of ATP, 12 kilocalories. So, okay, so 686 means 686 by 12 is equal to 12 5s are 50 and 86. Wait, somewhere around next ones are so something around this. So the round off number is this one. This is the answer. Okay, so we are done to some extent some of the questions. So now let's go to. So I'll finish this and then I'll leave you by 4 o'clock for the break. So respiratory substrate. You know that respiratory substrates are alone glucose. They are different substances. So any carbohydrate, everything, hexoses are the main one. Inulin, starch, these are all polysaccharides which get converted. Hydrolysis of these polysaccharide to glucose happens finally. So fat carry the most energy. So high calorie we say. So rate of respiration is high in the seeds, flower buds in dual region with respect to plants. With respect to animals, babies, infants will have rate of respiration more because of the metabolic rate is high, demand of oxygen is high, energy is high. Okay, so here 3 boxes in diagram represent. Okay, so I have, please solve it. So 3 boxes in the diagram, bio pathways, arrow mark shows. So pathway, glucose to pyruvate, pyruvate to, so find out. This is a beautiful question which I found. So this is not a neat question. I found this from an NSO book. Hopefully, you know NSO means. Are you aware of?
Yes, ma'am.
Yeah, so I found that as a very nice question. So do you know the answer? Do you know the answer here? Arrows represent net reactants or products. 4, 8 to 12. 4, 8 to 12. What are those arrows represent? That's what they're asking. So here arrow 4, 8, and finally 12. What are they? So I want you people find out the numbers from 1 to 12 basically. 1 to 12, what are they? What is 1? What is 2? What is 3? You should be finding. But here 4, 8 to 12. What is that 4, 8, 12? This is pathway A, pathway B, pathway C. What's that? Entry of glucose. So entry of glucose resulted. So what exit? What exits from that pathway 1? So now pathway glucose finally enters into pathway 2. What is that? How it is 2? So what are the different ways it to go here? So what is formed here and here? Water is formed and here what is? Anybody? Could you out? What is that we have forgot? Balance sheet we have got because of ATP formation. So this is ATP is formed everywhere. Am I right? So ATP is formed everywhere. Pathway 1A is glycolysis, B Krebs cycle, C is the oxidative phosphorylation. So therefore find out the other numbers. This is a very nice question. What is EMP here? 40th question. Anybody, can you give me the 40th question? What is EMP? What is EMP? Purvi?
Glycolysis.
Why EMP we call it? Emden-Meyerhof Parnas pathway, I told you this. So EMP can produce a lot of. So here appropriate is 38 ATP. Okay, so that's all. This is not there. So respiratory quotient. What do you mean by that? Volume of carbon dioxide evolved per volume of oxygen consumed. Okay, so respiratory quotient of carbohydrates. When we write the equation for respiration, so that there is a complete oxidation of glucose. So therefore, 6 molecules of glucose, so that is carbon dioxide evolved per oxygen consumed. So we write this side also 6 O2 and 6 CO2. So therefore, it is equal. So respiratory coefficient of carbon dioxide and oxygen is 1 here in this place. So with respect to fat. So this is a tripalmitic acid. So that is a fatty acid, one of the fatty acids we have taken as an example. So here 145 oxygen, for that 102 carbon dioxide given out. So it's 0.7, which is lesser than 1. So respiratory quotient, so incomplete oxidation. Here also, incomplete is lesser than 1. So therefore, complete oxidation happens only with respect to carbohydrate, and hence is change of carbohydrate. So there are how many questions here? 40 and here 41, 42, 43, 44. So I have given you almost 50 questions here in this. So this is made for NEET fast track actually, but I'm using it for your teaching also. So what is RQ? Respiratory coefficient is amount of carbon dioxide released per oxygen consumed. So that is CO2 by O2. CO2 by O2. Okay. So the respiratory quotient is the ratio of CO2 produced per O2. So to see different way it is given out. So apparatus to measure rate of respiration, which is that? Respirometer, manometer, which is that? Which apparatus is used? So this is with respect to breathing also. Can you tell me? Which apparatus is used? Respirometer to measure respiratory coefficient. You can put respirometer on the other window and see the picture of it. So it has a graduated tube attached to here, a bulb-like chamber. So respirometer here we keep plant twig. So we are talking about respirometer with respect to plant. So this is respirometer. So this will have a twig is attached in the bulbous. So this will be like this, a graduated tube. Respiratory quotient value of tripalmitin is, just now we discussed how much tripalmitin. So this is because of NEET point, we have taken this. How much is it? How much? 0.7, we just now discussed, no? Tripalmitin. Here, see here, 0.7, 102 by, so it is 0.7, you can write down. So this much is about the respiration plants. So we are done with this lesson. So anything you want to explain it again, please think about me before I go to the lesson. So I would like to cover the next lesson now. So after this, your exams are all over. All your exams over? No, I'm asking, your exams over, right?
Yes, ma'am.
Yeah, so shall we take up? So this is a 1 hour more than enough for me to cover this lesson. This is plant growth and development. So nothing required out of this NCERT for this. So we can take up that lesson now. That's what I thought of.